A plan talking about the gene drive and the vector surveillance using drones. So, Emmanuel, you have 5 minutes to summarize to us your key messaging. Okay, Karibu.
Okay, thank you very much. My name is Emmanuel, and I'll be sharing with you our work on the use of machine learning and infrared spectroscopy for rapid assessment of key entomological indicators of malaria transmission. So our stress of surveillance as the global technical strategy for malaria elimination recommends that countries should now integrate malaria surveillance into key intervention. And this means having a surveillance system that can capture essential malaria data in a complete, accurate, and timely manner. Example of these entomological parameters that we were talking about is the proportion of infected mosquitoes in the community, the preference of mosquitoes to bite human or other vertebrates, as well as the prevalence of malaria in the community. We measure these parameters using techniques like dissection, ELISA, and PCR, but these techniques are time-consuming and as well as requires expensive reagent. This is why we are proposing the use of infrared spectroscopy, which measures the chemical composition of a sample such as chitin, lipids, and proteins, which corresponds with either the age of mosquito, the type of host blood, or the infection status, or the resistance status. We have several examples that we have demonstrated this in the lab. They use a machine learning to distinguish 4 different blood-based hosts, achieving up to 98% accuracy. But to put this into practice, we performed this in the field setting, where we also achieved up to 90% accuracy but it's very challenging to get enough data to train machine learning models by doing field surveillance. So we try to build this from the lab and use that information to update the variation of the field information by using an approach called transfer learning. And you can see by estimating, using our machine learning model, we can estimate the human blood index with a good estimate matching the PCR. We have also demonstrated that on sporozoite detection, using ELISA and PCR as the ground truth to train machine learning models. As you can see, the difference between infections and non-infections is very minimal, and that's why we need machine learning to disentangle these small differences. As you can see as well, we can achieve good accuracy in distinguishing non-infectious and infectious mosquitoes. And also, we can estimate entomological inoculation rate using this technique as compared to ELISA. And you can see the estimates are not that much different from the main gold standard techniques. And the differences that we are detecting here are coming from the lipids, proteins, and carbohydrates that are associated with the parasite. We have also demonstrated this in detection of human blood, of parasite in human blood. And what we are now looking at is to assess the threshold of detection, as is already set in PCR and techniques like rapid diagnostic test, and to see how this technique will perform in areas with high and low transmission. We have also demonstrated using machine learning in distinguishing ages of mosquito, which is very important to assess the impact of intervention, as well as to distinguish between different Afrotropical malaria vector species. Now that what we are looking forward is there is this online platform that we are developing so that it can help this technique to be integrated in different malaria surveillance programs, to be able to predict the age of species, making this a one-stop shop to predict species age, infection status, blood sources, as well as parasite surveillance. Thank you very much.
Excellent. So Brian had prepared a 10-minute presentation. I told him 5, he used 4. So Brian, you can wait. We will have some questions. Now I welcome Saidi Jongo. You have 5 minutes strictly. You don't borrow the 1 minute from Brian.
Thank you so much. Presentation, please. Hello everyone. My name is Joe. I'm working with the Fakara Health Institute. I'll be talking about monoclonal antibodies technology. In principle, this is similar to the vaccines. There are two words, monoclonal antibodies. So the monoclonal is a technology. Antibody is not a technology. Antibody is what we get when we are vaccinated. In this case, we use the technology not really to yeah. Is it okay? Yeah. So in this case, we use the technology in order to administer the antibodies directly. This is the process that is usually happening when we are in the wombs of our mothers. We get these antibodies transmitted directly through the uterus, and that's how we benefit. That's how we get early protection from malaria. Otherwise, when you are born, you can no longer get these antibodies directly from your mother, because you can get them by breastfeeding in the first few months, but after that, you need to be vaccinated so that you produce antibodies yourself. The monoclonal antibody technology, meaning that we use the monoclonal cells in order to produce these antibodies, and they are administered by injection to you. So this is basically the other version of immunization. Basically, this is what we call passive immunization. If you get a vaccine, this is called, a few years ago, now we are here. What we believe is that with the technology that is currently available, we'll be able to take the monoclonal technology forward within the next 3 to 5 years to make it an application that is can be used in our communities. And therefore, first of all, there are already products that are being assessed. One of them is 643LS, which is updated to something called L9LS. This is manufactured by the NIH in the US. Our role into this is basically the validation through clinical trials. This is our value chain, meaning that we validate the product so that they can become application. In the same way that we validated vaccines, RHSS and R21, and now they have been approved by WHO for use. Meaning that we do the validation and they become application. Our target is also to do the same with the monoclonal antibodies. It has already undergone some early studies in the US and also in Africa and Mali. Next stage we are going to take it forward in order to bring it for several indications, including the use in pre-Malaria, this is also all the pregnancy levels, the use in infants and children, all age groups for people who, due to some immune compromise, cannot produce antibodies even when they're given a vaccine. We have evidence of that we have done ourselves. We have seen that there are population, for example, HIV positive, that when they're administered with the vaccine, their immune system is compromised and they cannot really generate the antibodies for future protection. And the monoclonal antibody will be one of the very important interventions in such groups. This is just the plan for the next 5 years. MAM01 is also another potential monoclonal antibody that is produced by Bill and Melinda Gates. And the ones that I mentioned by the NIH. I think these are just the concerns and potential solutions that we can discuss on the side after the presentation, and these include the safety issues, the cost of production, and also the manufacturing processes and the opportunities that are here in Africa that we can be involved. And generally, the technology requires multidisciplinary commitment, and I think we are doing that right now. Thank you so much.
Perfect. These guys are experts of the rapid fire. So can you fire the crabs for them? Perfect. So next is mmeelewa mjaelewa imeisha. Ni monoclonal. Sawa. So next now is a Brian Tarimo. You'll give us 2 talks. One will be on gene drive, and the second one will be on vector surveillance. So Brian, ukimaliza gene drive tuambia hapa ni mwisho wake, nimeanza vector. So you have 10 minutes for the 2.
Yes, good afternoon. Unfortunately, I do not have any slides, so I'll just speak. So I'll start with gene drives. Gene drives is a method of genetic biocontrol. What do I mean by genetic biocontrol? This is that you genetically modify mosquitoes so that you are able to control the disease they transmit. So mosquitoes can be modified. This historically, mosquitoes were being modified by a method called a SIT, sterile insect technique. But because mosquitoes, as any other living organisms, the method, the inheritance, follows Mendelian inheritance, meaning that you get 50% of the genes from your parent, your father, and another 50 from your mother. Now, if you sterile insect technique over several generations, that modification you've done by sterilizing these mosquitoes, you lose it. If I take this into the context of human beings, for example, let's say you're very tall, and you end up with a very short spouse, you might have short or tall kids. Now, if your tall kids continue meeting short spouses, eventually your tall gene will disappear. So if you take this into the context of mosquitoes, it's the same thing. You've made your mosquito sterile, but because there's 50-50 chance of inheritance, this sterility wins out, and over generations you end up with, you go back to zero. You need to introduce again sterile mosquitoes. Now what gene drive does, it biases this inheritance, the 50-50 inheritance, which is Mendelian inheritance. So instead of the tall gene I've been talking about, instead of 50% chance, you end up having 100 or near 100% chance. So if you sterilize your mosquitoes, over generation, this gene continues to appear in your mosquitoes. So gene drive is not, so gene drive is a means of passing this genetic modification throughout your mosquito population. Now, how do we do this genetic modification? There are two ways you can do the genetic modification. One is called genetic population replacement. That means you target the sterility gene in either all your mosquitoes cannot reproduce, then you have male sterile, so after generations, if you have all male steriles, all females are not steriles, after generation you have no mosquitoes. Another way is that you introduce a gene that kills the parasite inside the mosquito. We all know that mosquitoes are now born with plasmodium. They are vectors of plasmodium. That means mosquitoes will have to bite an infected individual and pick up the parasite. This parasite has to develop inside the mosquito for several days. Then when this mosquito goes and bites another person, that's when it transmits malaria. Now you introduce a gene into the mosquito such that the parasite is not able to fully develop. So this mosquito, when it goes and bites another person, it does not transmit malaria. So with regards to gene drive, what have we done at IHI? One, we've ensured that we have capacity building, we have the infrastructure, and we also have the personnel. Scientists inside the institution are able, one, they understand the technology, and they're also able to work with it. Two, what we've done, we've also established regular meetings, regular interaction with regulatory bodies, and we've also inside our institution formulated required regulation and required committees that are essential in order to work with this technology and to handle it. And lastly, we've also done several community engagement with communities where we envision or we plan that we think where this technology in future, when it's ready for deployment, it will be implemented. Vector surveillance. Now, vector surveillance. So throughout the day, we've been hearing from the National Malaria Control Program and other presenters that we need to do survey to see what kind of vectors we have. We need to know what type of mosquitoes we have that are transmitting malaria. We need to know if they have developed any resistance and so forth. One way you can do this, you can survey very competent entomologists. They can look at mosquitoes and tell you this is an opposite. They can tell you this is a certain type of mosquito, this is male, this is female. But if you want to go deeper to understand the molecular mechanism or the molecular makeup of these mosquitoes, you have to do some tests. You have to do some experiments. And one experiment you have to do, you can use next generation sequencing. That means you are able to get the entire genome of these mosquitoes. So there are standard genomes that are already published in several databases. If you do regular surveillance, you are able to identify what is the genome of these mosquitoes that we are collecting all over Tanzania, mainland and Zanzibar. Are they developing any insecticide resistance? This will be able to inform national malaria control programs if the current insecticides they are using are working, or if the mosquitoes have developed any resistance. We need to make programmatic or policy changes and so forth. So with surveillance, you can do two types of surveillance. You can do what we call amplicon sequencing, meaning that you already know the resistance, the gene that's causing the resistance. So you just go around, you collect your mosquitoes, or you look for it. But you also you can also do whole genome sequencing. That means you can sequence the entire genome of mosquitoes. This will enable you to predict or determine if there are any new these genes, insecticide resistance genes, species, and so forth. Now the challenge with sequencing is not the actual sequencing. Many institutions in Tanzania, including us at IHI in Bagamoyo, we have a sequencing machine. The challenge is once you have the data, what do you do with it? And with this, you need to have very competent computer scientists in computational power, because a sequencing machine generates a lot of computational data. So the analysis that's subsequent after the sequencing is the challenge, and this is something that us as IHI we are working to be competent in this area. Thank you very much.
Excellent. So, for those people who did not know exactly what does rapid fire session mean, this is the one. Okay? So Brian, you used 70% of your time. Another round of clap for Brian. So Brian, Jongo na Emmanuel, naomba mje hapa mbele. Naomba sasa kama mkuelewa gene drive. Kama ukuelewa kwamba ukiendelea kuwa watu wafupi, ile mbegu ya watu warefu kwenu itapotea. Ndio maana ya gene drive. Yani in the coming years, the only negative consequence of a mosquito biting you in the coming years. So that's what IHI is doing. Emmanuel, maswali. Any question for clarification? Question for clarification? 3